is on, my talk is on DNA testing in the popular media. So things that you may have heard about through different methods of consuming popular media, and what it actually means whenever people say they run DNA tests. So you can know how much is science and how much is fiction whenever you're watching TV. So just as a bit of a background as to my experience with DNA testing, so my bachelor's degree is in biochemistry with a concentration in forensics. I worked briefly with the FBI and then worked at the Arkansas State Crime Lab as a DNA analyst. And you know that forensics is extra hard in Arkansas because there's no dental records and all the DNA is the same. After I worked there for a few years, I got my master's degree also in biochemistry, and currently over at LSU Health Sciences Center going for my doctorate in biochemistry. So most of what I'll be talking about regarding DNA testing is from my experience with the crime lab, so if you have any questions, I should be able to answer them. So what we're going to cover is types of DNA testing that you may be familiar with. So some of you may be more interested in different parts of the talk than others. So first, if you watch a lot of crime dramas, we're going to be talking about forensic analysis. So if you watched a lot of NCIS, CSI, Criminal Minds, whatever. If you watch a lot of daytime talk shows, you may be interested in learning how paternity testing works. And if you pretty much watch anything else, you'll see commercials for Ancestry DNA testing kits such as Ancestry.com, 23andMe, and so on. So we'll go through how those are similar, how those are different, and what each different kind actually tells you. But in order to be able to understand what those different types of DNA testing are going to tell you, you need to make sure you understand what DNA is. So we're going to go just a brief review of what DNA is. And so what DNA we're going to start with, oops, come back. Okay. So there's a lot of different ways in which DNA can be described. So at its most fundamental level, you have these what they call base pairs, and they're referred to as A's, T's, C's, and G's. And what those are just abbreviations for the names of the chemical structures that actually bind to each other in the middle of the DNA. And because of their size and their shape, A's pair with T's, and G's pair with C's, and so this is how we figure out what the code of the DNA is. And so it's like an alphabet that only has 4 letters. And if you put enough of those letters together, like with any word, any letters, you would get words, and then you would get sentences. Sometimes you get a sentence, sometimes you get a paragraph, but once it turns into like an instruction for something, such as how to make a particular protein, that's what we call a gene. And we have different versions of genes, some that you get from your mom, some that you get from your dad, and we're going to talk about why the fact that you have different versions matters when it comes to DNA testing. So you have DNA, if you put several, letters together, you'll get a gene. Now, if you wrap all that up really tight so that you can package it inside a cell, you get what you call a chromosome. Now, a chromosome is just it's a structure that is how DNA is stored. So basically, it's like if you took the sentences and the paragraphs that are genes and you put them all together, you would get a book. Actually, you would get 23 books because there's 23 pairs of chromosomes in your cells. And so it's like How to Make a Human, volumes 1 through 23. And so that's what we're going to be looking at. So when people talk about chromosomes, what they're talking about is, one of those volumes. So you may have heard the, kind of unifying statement that all of our DNA is like 99.9% identical, so there are so few differences that should divide us. While that is a wonderful sentiment, it doesn't really help when it comes to forensic DNA testing and convincing people that you can tell people apart. So why don't all humans look exactly the same? Or why don't we look the same as our brothers and sisters if all of the DNA that I have could have only come from my mom and my dad, same as my brothers? So the way that works is a way, and this is a bit scientific, but we're just going to talk about how genetic diversity kind of arises with the idea that when you start out with a cell, you have two versions, like I said, of each volume of How to Make a Human. And when your cells are going to divide, they make a backup copy. So say you've got, this pink version from your mom's, this blue version from your dad. We're going with gender stereotype colors. And whenever you're going to copy a cell, you need another copy of that DNA that you can give to the other cell. So it's easiest to think about this so you have your original copy of the gene, and say it's like a hardbound book. But when you make a copy, you make it as a binder. And sometimes, you ever dropped a binder and it pops open and all the pages just fall out everywhere? So you have two very similar copies of these sets of instructions for how to make a human. And say you know enough context clues to put everything back together, but if the sequences are the same enough, you can put things in the right order, but sometimes you'll put a page from your dad's binder into your mom's binder by accident. And so the pages will get shuffled. All the instructions are there, but they're a little bit different. This page used to say blue eyes, now it says brown eyes, because that's what the other version said. And so little bits of the DNA get exchanged between the chromosomes. And so whenever you're going to make eggs and sperm, which we're going to have to combine later into a new person, these get allocated into different gametes. So the cells, originally they split into what they thought was just your mom's copies and just your dad's copies, but in reality, some of the pages, some of the sentences or paragraphs have been exchanged. And so you end up with where each cell, each egg or sperm, has a unique copy. And so that's why you don't end up looking like your brothers or your sisters, and it's this sort of recombination that makes both genetic diversity and many different kinds of DNA testing possible. So first we're going to talk about forensic DNA testing, mainly because it's my favorite, it's what I have the most experience in. Now what forensic DNA testing is comparing an unknown DNA sample of forensic relevance to a known sample. And that's really all you're allowed to do. So what you're doing is you're pairing something that you found at a crime scene, and you're comparing it to a known, and the known can be either the victim, a suspect, a relative, basically anyone who could have had cause to leave DNA in a specific area. So what you're looking for is a specific type of DNA called short tandem repeats, and I think this actually got mentioned earlier in the East talk. So what a short tandem repeat is basically it's a little chunk of DNA that gets repeated, and those repeats are right next to each other. So literally that's what that means. But what it is basically is like if you have a word and that word gets repeated a variable number of times. So if this repeat here, this means that if in this section of the How to Make a Human book, this sentence reads, beer is very, 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 very tasty. This version, because everyone has two versions of each gene, says beer is very, 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 very tasty. Only 6 copies. So if you're looking at the number of repeats, you would call this a 7, this a 6. And the other person would have a 4 and a 19, a 12 or a 14. So these are highly variable regions. If you notice, this doesn't tell you how to make a human. This is just like a statement that isn't an instruction for anything. And the regions of DNA that you look at in forensic testing are not descriptive. So when you get a forensic profile, you don't look at it and say, oh, this came from someone from Utah. I mean, whatever. But it doesn't tell you anything about the holder of that DNA. It doesn't tell you their height or their gender or their race. Well, it does tell you their gender, but it doesn't tell you their race or anything like that. What it does tell you is a readout like this. So you put the DNA into a machine that separates the repeat segments based on size. And so the more repeats, the slower it runs through the machine, and it comes out, and it just basically, based on how long it took that DNA to run through the machine, tells you how many repeats there were. And so you just call it based on size. And this is an example of a forensic DNA profile that I just pulled off the internet. It's not anyone I know. So you look at these 16 different areas, 13 of which are chosen by the FBI and every crime lab in the country needs to use. And then the other 3 are one that tells you XY or XX, so is it a male or female? And then you can kind of choose the other 2 based on personal preference, basically. And what you do is based on the frequency of these different, how often does someone have a 15 at this gene? How often does someone have an 18 repeat at this section? So you do the statistics on that. You say, how likely is it that just some random person would have this profile? And if you have this profile in a forensic sample, and then you have that profile from a person that you know who they are, you calculate how likely is it that this just randomly happened? Barring an evil twin theory, how likely is this? And in order for a DNA analyst to say, with scientific certainty, these two profiles match, you have to, the minimum threshold is that you would need 50 Earths worth of people to have another random match. So, and that's the minimum, and most samples far exceed that minimum threshold. But, like I said, barring evil twins, you need 50 entire Earths worth of people in order to find another random match just completely by accident. And so that's what forensic DNA testing tells you. It doesn't tell you this person committed a crime. All it says is that statistically, it'd be unlikely for these profiles to just randomly match. So if you watch a lot of CSI or NCIS, you may have some lofty expectations for what forensic DNA analysis can actually tell you. Your minimum turnaround time is going to be, at least a few days. Given a backlog, it's going to be a few months. It's not going to be, 60 seconds in a scene while someone paces nervously and then, their driver's license pops up on the screen next to you. Not how that works. But all it can tell you is whether or not these two profiles statistically match. Very similarly related is paternity DNA testing. So what that is comparing an individual's DNA to the suspected relative's DNA. So your general options are going to be someone either is a father or is not. This can be done for mothers, but generally there's less uncertainty about whether or not someone gave birth to a child. It happens. So one version of paternity testing is a lot like the one we just talked about, where you generate that profile where you have, two versions of, the short tandem repeats, how many words there are, the length of the DNA segment. And so you get two versions from your parents. So this is the child here in the middle. So you know that one of their How to Make a Human copies came from their mom and one came from their dad. One of those had a sentence that had 23 repeats in it. The other had 24 repeats. Now, if this is the mom, you know that the 24 repeat is the one that came from her, because she could have only given a 22 or 24. So that means the dad has to have a 23. The alleged father does not have a 23. So, not the father. As a fun aside, this is my family's profiles. I did this for my parents as a anniversary present for them, and luckily it worked out. When I gave this to my parents, my mom was like, "What are you trying to do? Get me in trouble?" But anyway, so you can see my father's profile here at the top, my mother's there, and these are like the 16 areas. I didn't label them, that's not very artistic, no one cares. But you'll see that for me and both of my brothers, every peak that we have representing a repeat of different number of repetitive segments, each peak will match either to my mother or my father, and I have color coded them accordingly. That's the kind of nerd presence I give. The other option is YSDR testing. Now, the bad thing is that only works for guys. But it is, there's less variability, because when you get those copies of How to Make a Human, there's one version that can't do that recombining bit, where if you shuffle the pages in the binder, is your X chromosome and your Y chromosome. For the most part, there's not a lot of exchange between those, and if only your father can give you the Y version, it's going to be a photocopy every time. So you only have one locus to compare. Statistics are easier. But the downside is that you can't distinguish between male relatives in a line. So like a male will have the same profile as his father, but also his brother, and his father's brother, and his paternal grandfather. So if there's ever any question in that realm, this kind of testing doesn't help you. But as far as like boy child to suspected father, it's often very helpful because, like I said, the profiles are less confusing. You don't have to consider the mother's profile, things like that. The last topic I'm going to cover in categories of DNA testing is ancestry DNA testing. You probably hear a lot about this either on the news or, like I said, just purely in commercial form. So this can be comparing an individual's DNA to profiles that are typical for a given geographical region, or it can also be referring to determining if individuals are related to a common ancestor. For this, we're actually looking at a different type of DNA. I mean, all DNA is still just A's, C's, T's, and G's, but we're not looking at how many times a particular word is repeated. We're looking for spelling changes, basically. And again, it's not changes that usually mean anything. Sometimes it is, and we'll talk about that whenever there's medical implications involved. But for the most part, this is a simple spelling change that are called single nucleotide polymorphisms, which means there's just one base that's a different spelling. And so whenever these tests like Ancestry.com, which I use as an example because they've got a good lot of examples online of how they do their data, and they'll give you an example of what their raw data looks like. They'll tell you like on chromosome 1, so volume 1 of how to make a human, they look at, on this page, on this line, on this letter, you expect to find an A in one of your copies and an A in the other copy. And then you do that at 700,000 different places in How to Make a Human, the box set. And they compare your DNA to what they call native populations. And basically that just means that someone that can prove that their family has lived in that area for like 300 years. And that's what they call, that's what they use as their reference population. Current residents in an area may not necessarily be, quote, native to the region, so that's why they require you to prove that your family is representative of the genome in that region, and that's what they compare you to say, oh, your family's all from Switzerland. Weird. And so that's what they use, what they've established as native population. So whatever ancestry testing site you choose, whether it's Ancestry.com or MyHeritage or FamilyTree.com or what, like there's a ton, but the benefit is based on the size of their database, because the more they can compare to, the more accurate your results will be. You can also, like I said, you can use this to compare ancestors to one another, like compare people and what their last common ancestor was, or you can say like, a person who is alleged, you can use a person who is a descendant of a known individual in like these two examples, which were, they found the grave of Richard III, great, and then they found another grave that everyone thought was Louis XVII, and they're like, no, sorry, not that guy. So this is like cool historical discoveries that have come about using modern scientific developments like DNA testing. You also may have heard that 2014, I think the FDA said it's okay for them to tell people like risk factors that they may have, like popular mutations that may lead to cancer or other degenerative diseases. But the only diseases or conditions that they can speak to are ones with very well documented genetic causes, or ones that involve very specific changes. So if you know that this one change in this one letter is going to predispose you to sickle cell anemia, that they can speak to. But most of your traits are far more complicated than that, and they can't give you a ton of really definitive data about your medical conditions. Very last thing I'm saying, just because this was a very interesting thing that just happened last month and was putting a lot of combination of forensic DNA testing and ancestry DNA testing into the news, was the identification of the Golden State Killer. So this is actually, and at first I was like, how can you like the types of DNA testing that you get from forensics versus an ancestry site are very different. Like I said, the repeated words versus a misspelling, basically. But there was a paper that came out in 2006 about integrating patterns of polymorphism as single nucleotide polymorphisms and short tandem repeats. So, what they do is they actually tell you how to convert. You're not going to get like a letter-by-letter profile of an ancestry readout based on short tandem repeat data, but what it will tell you is the likelihood of someone with this ancestry profile being related to someone with this forensic profile. So what they do is you look at these two sentences, his shirt is a very pale gray with an A, or his shirt is a very, very pale gray with an E. And so this is an example of STR data. How many times does this repeat? This is an example of short nucleotide polymorphism data. There's one letter that's different. Meaning didn't change though. So what you're interested in how they do these calculations is you find the odds of having one vary and gray with an A, or one vary with gray with an E, or two varies and gray with an A or D. And so basically you're just calculating the odds that someone with one vary would also have gray with an E. So that's how you would relate those two. And like I said, it doesn't give you a definitive profile, but it can put you in touch with a likely family that someone with that forensic profile is related to, that then they can investigate further and actually get the proper type of DNA data that you're looking for. And so those are the types of DNA testing that I wanted to cover. And if you have any questions, please let me know. Thanks for listening.